Hello, everyone, and welcome to this new webinar entitled Optimizing Drug Discovery Pipelines with Biophysical Methods. This webinar was made possible through sponsorship from Wuxi AppTech. As always, I'll be your host. I'm Jeff Pugaliskas, Technical Editor for Genetic Engineering and Biotechnology News, which is celebrating its 40th year, bringing you the latest information on advances within the life sciences industry. Drug discovery is an invaluable part of the development process for therapeutic compounds, and in the past several years, researchers have been desperate to find improved biophysical technology that can streamline discovery methods. Technologies such as surface plasmon resonance, NMR, microscale thermophoresis, isothermal titration calorimetry, and dynamic light scattering play an essential role in studying binding affinities, kinetics, and thermodynamics for lead molecule or target identification. These advanced techniques help improve speed, sensitivity, and throughput, allowing for the biophysical screening of thousands of small molecules in a short time. And today's presenters are going to tell us in more detail about the application of these biophysical technologies to support drug discovery programs. So let's meet our speakers and we can get to their presentations and find out more. Dr. Moran Jerebic is head of biophysics and screening at Wuxi AppTech and will lead things off by providing us some background into biophysics and screening methods, as well as dive into a specific case study to show the application of some of these techniques. Moran will turn the presentation over to Dr. Annika Neidner, who is the scientific lead for biophysics and screening at Wuxi AppTech, and will describe for us technology used for fragment screening as well as antibody binding kinetics. And then to wrap things up, Annika will turn the presentation over to Sunling Bing, who is a director of biophysics at Wuxi AppTech and will tell us more about new techniques for small molecule discovery. But quickly, before we begin, I want to remind the audience, as usual, to stick around for the Q&A session that will occur right after the presentations end. If you have a question, you can send it in at any time during the presentation. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. Okay, so we hope you send in all the questions that you may have. And with that, Moran, I'll turn things over to you.
Welcome to the webinar, Optimizing Drug Discovery Pipelines with Biophysical Methods. The seminar or the webinar today will focus on state-of-the-art biophysics platform at Wuxi AppTech and on biophysical solutions to support challenging drug discovery programs. My name is Moran Jerebic, and I am leading a team of biophysics in Wuxi AppTech. I will give you general introduction to biophysics and screening capabilities in Wuxi AppTech, and I will introduce you a case study showing how we were able to establish assay for a very difficult drug target called NLRP3 involving inflammation. I brought with me today two very experienced biophysicists, Dr. Annika Neidner and Dr. Sunling Bing. They will share with us how biophysics can apply for fragment screening and orthogonal validation of their hits, and how binding kinetics of P3 specific antibodies was used and performed on Biotor 8K, and how we can apply biophysics to discover Bifunctional small molecules, similar as protein development by Dr. Sunling Bing. I am very excited to be here today. I hope you will find this webinar useful and interesting, and I'm looking forward for the Q&A session. Save your question, put them in the chat, and we will be happy to discuss it together with you during the Q&A session directly after this presentation. In Wuxi AppTech, we support clients and partners around the world in giving biophysical solution for different needs. Biophysics is highly important at any step of drug discovery. So when we initiate a drug discovery program, we typically want to show that a certain small molecule that we hope to identify will bind a certain target protein. Therefore, a prerequisite for drug discovery is to have a high-quality protein, which can be used in biophysics. Therefore, the first step before initiating any screening is to see the quality of our protein, since we want to have proteins which are functional. So at least we want to show affinity to a certain tool compound, substrate, or any other well-characterized and established binding partner. And we want to have a monodispersed protein which do not aggregate. And all of this can be tested during assay setup. After development of an appropriate protein, which is functional, do not aggregate, is monodispersed, and well folded, we can establish screening, for example, as fragment-based screening or SMS using different biophysical methods. After the screening was performed, we have identified hits. And these hits need to be validated using different biophysical methods. Of course, we identified our hits and validate the hits that we have identified, but sometimes clients ask us to validate hits which they have generated in-house or by another partner by coming out of fragment screening or HTS, in silico screening, or from a DEL screening. We will hear later today more about DEL screening, a very powerful technology which allows to screen millions of molecules in a single tube in a very short time. And the best or most promising DEL hits need to be biophysically characterized and validated. After hit validated is completed, we hopefully have identified a few lead molecules, and these need to be further characterized by measuring binding affinities, binding kinetics, binding energetics. It's always very important to understand where the molecule binds, so mode of action studies play a very important role as well. So as you can see and recognize in the slide, biophysics is very important at every step of drug discovery program, starting from target to hit, moving to hit to lead, and during lead optimization. And in many cases, biophysics is still important at a later time point as well, as a compound is still sometimes further modified and optimized. In order to give solution, biophysical solution at any phase of the drug discovery program, we have established a very versatile toolbox containing different biophysical and biochemical methods and assays. Here we can see an overview for the established biophysical and biochemical platform allow us to give you best-in-class solution for your drug discovery program. So in our toolbox, a very versatile toolbox, we have access to methods which allow to measure binding, thermodynamics, stoichiometry, protein quality, kinetic, and function of your proteins. The different method has different sensitivity and throughput. So with the available methods, we can run low number of compounds up to 20 to 50,000 compounds. So in the methods that are available, we have methods which in HTS format as well, like Bio48K and the Dianthus. And if I'd like now to spend some time and go and discuss the different technology which are available just in short. So SPR is one of the core technology in WuXi AppTec. In our WuXi-AppTec Biophysics platform, we have multiple number of Biocore 8K and 8K plus. With this technology, you can measure binding affinities and binding kinetics between protein and small molecules, for example. And it has pretty high throughput, so we can run screening of up to a few thousand molecules a week, or just profile a limited number of compounds depending on the need of our partner. In another technology which play a very important role in compound profiling is microscale thermophoresis. In WuXi-AppTec, we have now over 10 years experience with MST, and MST was, or WuXi-AppTec was the first CRO worldwide to establish and to integrate microscale thermophoresis into our drug discovery pipeline. Meanwhile, we have characterized over 200 different protein targets just with MST and TRIC. The TRIC technology is a new device. I like to call it MST second generation. So it's based on the MST technology, but it allows us to measure binding affinities in free solution on a 384 plate. I just want to mention that since we are using the technology for over 10 years, I would say we are the most experienced CRO as well for microscale thermophoresis technologies. And ITC is another very important biophysical tool to measure binding affinities and binding energetics. The technology, we are mainly using it to profile and to understand our lead molecules, since the method needs a lot of material and has very low throughput. Other technology which play an important role in our WuXi-AppTec platform are the different QC methods to analyze protein quality and stability, like DSF, nano-DSF Panta, and DLS. Two other important screening technology are, of course, mass spec and NMR. Using fluorescence reader, we have established different fluorescence-based biochemical assay in 384 plate as ADP glow, for example. This very versatile toolbox allows to find multiple solutions to assess molecular interaction. And this very versatile toolbox gives us access for protein-protein interaction, protein-small molecule interaction, to run fragment screening, to work with membrane protein, to run binding of DNA or RNA to protein. We have a lot of experience with biologics as well. To understand mode of action by running competition experiments. In the last years, we saw a huge increase in the interest in biophysical characterization of binary and ternary complex formation. For example, characterization of Bifunctional molecule as protein and molecular glue. This can be done biophysically very nicely. We have established a very nice platform that Dr. Lingbin will show us later how a Bifunctional small molecule can be characterized. And it's really important for us to understand how ternary complex formation or assembly take place in solution. Regarding affinity range, we can cover affinity with the available technology from picomolar to millimolar. Now I'd like to start with the first case study for today, where we are going to study the binding affinity between NLRP3 and small molecule. What is NLRP3? NLRP3 is a very important protein, which is part of the inflammasome. Upon its activation, NLRP3 inflammasome induced or triggered inflammation-dependent programmed cell death called pyroptosis. And therefore, NLRP3 is a very hot target since it plays an important role in different diseases as Alzheimer's, atherosclerosis, asthma, and other allergic inflammation-related diseases. The main challenge with NLRP3 that, as you can see, upon its activation, it's polymerized and it's aggregate. So this is actually what we really don't want to have when we do biophysics. Unfortunately, this is the function of the protein. Therefore, we have spent a lot of effort and a lot of time in order to find a solution that will allow us to run biophysical, biochemical assay with NLRP3 protein. And after putting a lot of hard work, a lot of resources, and after in total analyzing and trying over 70 different constructs, we were able to produce with high protein yield and high quality and activity validated assay grade protein. Here we can see the analytical set, which show a very reasonable homogeneous peak, and the protein can be used in different biophysical and biochemical assays. In order to test the protein is well folded, we run nano-DSF experiment. We were happy to see the relative sharp unfolding. What is really nice to see as well, that the protein, which is an ATPase, bind ATP. In the presence of ATP, we see a clear increase in its protein thermal stability. In the presence of ATP, the protein bind MCC950, which is one of the most developed NLRP3 inhibitors. As a next step, we decided to establish binding assay to allow our partner to measure binding affinity between compounds and the target protein. And as you can see here, the results of 3 compounds, which I allow to show to you. All of these are commercially available compounds. Here we can see the binding fluorescence level NLRP3 to MCC950, oridonin, and Bi11. The signal to noise is really fantastic. So that's actually comparing to what we found in the public domain, the most impressive NLRP3 assay available right now. And we able now to use this assay to run screening or validate hundreds of thousands of small molecules. In addition, it is really nice to see that MCC950 induce a very different MST response comparing to oridonin or Bi11, which bind to a different binding site and have a different mode of action. Oridonin is an covalent inhibitor. After establishing binding assay, it is important to test if this compound only bind or can they affect the ATPase activity of the protein. Therefore, we establish an ADP glow assay. On the left, you can see the Km for ATP, which we have determined to be at 20 micromolar. And on the right, you can see very nicely the inhibition of the ATPase activity of NLRP3 by MCC950. In order to summarize, you can see that after putting a lot of efforts, we were able to establish a ready-to-go assay to support our client in profiling small molecules against such complicated and challenging target as NLRP3. In the next 2 case study, Annika will tell us how biophysics is applied in order to run fragment screening. She will introduce the workflow for fragment-based drug discovery in WuXi AppTech and will show us how DEL hits can be validated using different biophysical assays.
So thanks for the kind introduction. So I will start with fragment-based drug discovery. What is fragment-based drug discovery? So in fragment-based drug discovery, a diverse set of 3,000 fragments represents its chemical space about as effectively as would 10 trillion diverse drug-sized molecules. These fragments bind to small pockets available on the protein surface, and therefore it is ideally suited for targeting protein-protein interactions and challenging targets. High hit rates of 3 to 10% are an excellent predictor that high affinity small molecule ligands can be identified. These fragments often offer unique binding profiles with the target. And through chemical optimization, we have the ability to optimize pharmacokinetic profile simultaneously with potency as the fragment hit grows to a clinical candidate. How does FBDD workflow look like? So this is actually a typical workflow that starts with a fragment screen as a single point screen using either MST or SPR. The hits are further confirmed and KDs are determined using the very same screening technology. Subsequently, we have an orthogonal hit validation using, for example, either SPR, MST, or nano-DSF. And this is then followed by structure generation for the hits, either by X-ray crystallography or NMR. Depending on the outcome, one can either have a structure-led fragment to lead development or an activity-led fragment to lead development. How does the workflow, the FBDD workflow at WuXi AppTech looks like using MST, TRIC, and SPR? So actually we have 3 building blocks. And since a prerequisite for a successful FBDD is a high protein quality of the tested target, we implemented here a protein production and the protein QC, which allows us to have various constructs. We offer monodispersed protein that is highly pure. We are able to do buffer optimization, an activity check, and batch-to-batch comparisons. The second building block is then the actual assay setup and primary screen, where we use our in-house WuXi AppTech hits fragment library of 3,100 fragments that we can screen with MST or SPR. And the hits are then validated and also orthogonal validated and KD ranked in the second building block. And this is then followed by a third block, which includes the structure determination with a co-crystallization. We have a so-called system for hit validation by crystallography in hand. And the next building block is then the structure-based drug design, where we can also support within the WuXi AppTech company. In the next slides, I would like to draw your attention to one case study where we have used an MST-based fragment screen for the sting. So what is sting? So we have used the human sting. Sting is a stimulator of interferon genes. It plays an important role in innate immunity. Sting induces type 1 interferon production when cells are infected with intracellular pathogens such as viruses, mycobacteria, and intracellular parasites. Type 1 interferon, mediated by sting, protects infected cells and nearby cells from local infection by binding to the same cell that secretes it and nearby cells. STING works as both a direct cytosolic DNA sensor and an adapter protein in type 1 interferon signaling through different molecular mechanisms. It has been shown to activate downstream transcription factors STAT6 and IRF3. In this slide, I will show you the assay development in the primary screen. So in the assay development, we have used a cGAMP as a tool compound. And you can see in the structure over here a homodimer of STING and a cGAMP binding to the dimer-dimer interface of this protein. Using MSD, we did the assay development and validated the assay and showed that the tool compound has an affinity of approximately 10 nanomolar. Subsequently, we performed the primary screen where we observed a hit rate of 8%, and we confirmed these binders with a KD validation, so a KD determination also using MST. In a second step, we went for an orthogonal validation, and here I will show you the techniques that we implemented. First of all, it's SPR. Here you can see the tool compound binding, yielding a KD of approximately 17 nanomolar. We've also used nano-DSF, where the tool compound cGAMP leads to thermal stabilization of the protein by more than 11 degrees Celsius. And we have used STD-NMR to validate the binding also in solution. So what is the STING study here in numbers? So initially we started with 3,100 fragments. We used 4 orthogonal assays and identified 66 binders. These binders have an affinity between 10 and 500 micromolar. From these binders, we identified 11 best hits, and 9 of these 11 best hits were then validated with STD-NMR as well. As you can see, fragment hits have a relatively low affinity for the target protein. However, chemical optimization of the pharmacokinetic profile can improve the potency and selectivity dramatically. And to underline this, I will show you an example where FBDD led to an FDA-approved drug in 2011. And here you can see that, for example, an early candidate that came out of the fragment-based drug discovery screen has a really low IC50 for a kinase. However, this compound was further developed. There was a chemical optimization. You can see the potency increases, and in the end, the FDA-approved drug over here has an IC50 value of just 30 nanomolar for the target kinase over here. So, and with this, I will already go to another screening technique, DEL screening. So what is DEL screening? I will give you a short overview. What is a DEL workflow? DEL stands for DNA encoded library screening. So in the screening, more than 65 billions of chemical compounds are conjugated to short DNA sequences that serve as identification barcodes. The target protein is immobilized to an affinity matrix, and interacting chemical compounds are enriched. Through sequencing and decoding, these hits are identified, and the hits undergo further hit validation. And in this webinar, I will focus on the hit validation we performed for a kinase DEL screen. And in this slide, you can actually see the assay setup for 3 biophysical techniques. For this wild-type kinase, our target wild-type kinase, and you can see the tool compound binding in SPR, yielding a KD of just 0.2 nanomolar. using MST, we identified a KD of 1.8 nanomolar. And in nano-DSF, this tool compound leads to a thermal stabilization of the protein of more than 5 degrees Celsius. We then screened the DEL hits, also using the 3 assays. And I will show you the results for compound number A, binding to the target wild-type kinase in the absence of tool compound. So for SPR, we identified a KD of just 0.6 nanomolar. And with MST, it's 86 nanomolar. And also in nano-DSF, we confirmed a strong binding with a thermal stabilization of more than 5 degrees Celsius. That is really strong. Of course, we were also interested whether this compound A also binds in presence of tool compound. And therefore, we added tool compound to our assays, and we did see that in SPR and MST experiments, there is a dramatically weaker binding. We don't observe saturation anymore. And also in nano-DSF, the thermal stabilization is weaker. It's just 1.8 degree. Besides wild-type kinase, we were also interested in the mutant form of this kinase. And therefore, we also did the assay setup for this mutant kinase over here. And you can see using SPR, we determined a KD of 0.2 nanomolar. For MST, it is 0.7 nanomolar. And also in nano-DSF, with this mutated kinase, we observe a stabilization and presence of tool compound of more than 7 degrees Celsius. Does this mutant kinase also bind this compound A? And the answer you can see over here. So in absence of tool compound, we again see a strong binding. This time it's approximately 2 nanomolar using SPR. With MST, roughly 20 nanomolar. And in nano-DSF, there's also a strong thermal stabilization visible. What happens if the tool compound is present? And again, you can see that there is no saturation visible in the SPR experiment and in the MST experiment, and also in nano-DSF. The thermal stabilization is weaker. So, to make a long story short, DEL screening is capable to identify lead candidates that bind to the target protein with nanomolar or even picomolar affinities. In comparison, fragment-based drug discovery yields initial hits that just have micromolar affinities. However, both screening techniques have advantages and disadvantages in terms of binding site accessibility, chemical optimization, selectivity, and potency. Therefore, it can be an advantage to combine both screening technologies for the identification of drug candidates.
With around 100 therapeutics antibodies in the clinic, development and characterization of complex therapeutic antibodies which bind to multiple targets is of high interest. Annika will show us how SPR was applied in characterization of B and 3 specific antibodies using Biophysics 8K.
Thank you very much. To underline the importance of bispecific and trispecific antibodies, I would like to draw your attention to this example for anti-cancer immunotherapy. So a trispecific antibody recognizes the myeloma cell depicted over here via the CD38 antigen. A T cell is brought into proximity via the interaction of the antibody with CD3 and CD28 antigen, which also activates this T cell then in the end to kill the myeloma cell. For all bi and trispecific antibodies, it is important to know whether the binding of one antigen disaffects the binding of other antigens, for example, through a sterical hindrance. To answer this question, it is necessary to perform a sequential binding experiments and kinetic measurements individually or with other binding arms occupied. The gold standard for such antibody characterization is SPR, and in this picture you can see the assay setup. So we have a CM5 chip where an anti-human IgG that recognizes the FC tag or the FC region of the antibody is immobilized to a dextran matrix via amine coupling. Subsequently, a B-specific or the TRIC-specific antibody is captured. This is then followed by the injection of the antigen 1, 2, or 3. And after each analyte injection, the surface is regenerated back to the amine-coupled anti-human IgG. And we will start with a case study now with the bispecific antibody and the sequential binding experiment. And here you can actually see 3 experiments. And the first one is depicted here in green. That's the binding of antigen 1 and 2 together. And you can see here in sensogram in green, this yields approximately an Rmax of 37 response units. In a second experiment, you have first an injection of antigen 2, leading to a response of approximately 15 IU. Subsequently, antigen 1 and 2 are combined in the next round of injection, leading to a response level of 37 IU. And in the third experiment, this experiment is inverted. So first we inject here in blue antigen 1, and subsequently a combination of antigen 1 and antigen 2. Injection of antigen 1 yields a response level of approximately 22 IUs, and then again A1 and A2 together, we can again reach a response level of 37 IU. So A1 and A2 antigen, they showed independent binding to this B-specific antibody. In the next step, of course, one has to look at the kinetics, whether they are also the same in presence of the other binding arm occupied. And this you will actually see over here. So on the left experiment, that's just the titration of antigen number 2. And on the right side, there is the injection of antigen 2 in presence of antigen 1. And if you can have a look here at the kinetic values as well as the overall KD, they're really similar to each other, indicating that they bind independently to each other. So there seems to be no sterical hindrance. In the next set of experiments, we performed a similar set of experiments for this tree-specific antibody. Here in green, you can see in the first sequential binding experiment, the injection of antigen 1, 2, and 3 altogether, reaching a response level of 47. In another round of experiment, we first injected antigen 1 and 2, reaching a max of approximately 25 IU. And this is then followed by a single injection of A3, where we reach approximately 42 response units. This already indicates that there is an independent binding of all antigens to the tree-specific antibody. However, we still want to know whether the kinetic is also not affected if binding arms are occupied. Therefore, we performed kinetic measurements. And I will just show you here one example. So that's the binding of antigen 3 alone. Here, for example, we observe a KD of approximately 0.4 nanomolar. And on the right side, you can see also the binding of antigen 3, but here the binding arms are occupied with antigen 1 and 2 as well. And you can see the overall kinetics, and the KD is also 0.4 nanomolar in this case. So based on this experiment, we can conclude that there is independent binding between all 3 antigens. And to conclude these experiments, I would like to point out that our WuXi AppTec biophysical platform is a powerful tool also to characterize biologics.
Thank you very much, Maren and Annika, for showing us how important the biophysics assays could work in different therapeutic areas. And I would like to switch the gear a little bit from bio or tri-specific antibody to another recently very hot topic, the biofunctional molecules. And I would like to use Cerebrum DDB1 as a case study to show you how we design the study and interpret the data to help us move the biofunctional molecule projects forward. And as you can see here, this is a very classic developed cascades for a PERTEC. And I would like to emphasize very importantly how cooperativity of the ternary complex could work in the structure-based rational design. As we move into the SAR stage, we have different assays, as you can see here, the biochemical, biophysics assays, as well as the cellular assays. But how we are going to use these assays to guide our studies and the developments. So one very important thing is we could get the cooperativity of the ternary complex formation over the binary complex formation using either biochemical assays or biophysics assays. So for biochemical assays, we could use TRIC, we could use alpha screen, and for biophysics assays, we could also use SPR and MST. And today I'm going to introduce more how we interpret or design the experiments using MST. And from the experiment data, we could calculate alpha value, and if alpha value is over 1, then we could call it with cooperativity, and it's a good indication of the compounds forming ternary complex more stably comparing with the binary complex, a very good sign for the compound to stabilize the ternary complex formation, and sometimes people thought it's a good indication to overcome the hook effect and better effect and degradation in the cellular. And using this, we could further combine with structure evaluation for the ternary complex, which all together could contribute to the structure-based rational design of the PERTEC. So how are we going to run this study? So we could start from the binary complex formation, which is a pretty routine and everyday use for the MST technology. So basically we label the protein using a TRIC specific dye, and then we mix the labeled protein with the biofunctional compounds, and then we could get the TRIC signal shift from where we could calculate the KD value for the binary binding. And for the ternary complex formation, it's a little bit more complicated, and we need to simplify the ternary complex formation still into more like a binary binding. By doing that, we will still utilize the labeled target protein and assay conditions from the binary interaction assay. And then we could pre-incubate the series dilutions of compounds with saturated concentration of the target protein or the E3 ligase, and then measure the ternary complex KD using the optimized conditions. Then for the alpha value I just mentioned, we could calculate the value by using the KD binary divided by the KD ternary. And if it's over 1, then it's a good indication of cooperativity. So taking DDB1 cerebrum as example, so as you know, CCA5 is emit compounds and bind to the DDB1 cerebrum and could induce the degradation of GSPT1. So here we could show that if we label DDB1 cerebrum and we titrate the CCA5 compounds, we could get a very nice MST curve and get the KD value at around 30 nanomolar. And for ternary complex assay, if we want to determine how the GSPT1 bind to this new ligand surface developed dedicated by these compounds, then we could pre-incubate the labeled E3 ligase, which is DDP1 cerebrum, with the saturated compounds, and then titrate the GSPT1. So you could see the two data here. If we add 5 micromolar CC85, then we could get a KD value of around 19 nanomolar, which indicates the KD of the GSPT1 bind to this new interface contributed by the compounds and DDP1 cerebrum binding. But if we include another cerebrum binder, we could detect no binding at all. So this data clearly show the new interface development by the compounds and the ligase. But if we want to calculate the cooperativity, we still need to titrate the compound instead of titrate the protein. So what we do here is still we have this binary binding for the CC85 to the cerebrum. And for the ternary binding, we still label the cerebrum protein and then titrate the compounds with the excessive amount of GSPT1. So then we could get a much lower ternary complex KD. And as you could see here, the alpha value calculated is 39.6, which is very high and indicating a very good cooperativity of the ternary complex formation over the binary complex. And this could indicate or lead to further study of these compounds, maybe in the structure biology or other studies, to better elucidate how the compounds trigger the ternary complex formation and induce the protein degradation. And by the way, this is the case for molecular glue, but for the PROTAC, similar principle could be applied.
Thanks a lot, Annika and LinkedIn, for your contribution. Now we are coming to the end of our webinar, and I'd like to summarize what we have just discussed and what are the key messages. First of all, orthogonal biophysical characterization is essential for versatile drug discovery programs and allow to get an almost full picture of the chemical matter at all stages of the development process. Here, binding affinities, binding kinetics, binding energetics play a very important role for in-depth compound profiling. So assay setup and optimization together with intensive protein QC are of high importance for the outcome. Feasibility studies during assay setup help to find the most suitable technology for the target protein. And now is the question, what do we hope to see in the coming years? Hopefully integrated and fully automated plate-based platforms will allow screening of much bigger compound libraries and will enable biophysical HTS. Biophysical screening in vivo-like conditions like MST, TRIC, and in cell NMR hopefully will open new possibilities. New strategies to purify membrane proteins in assay-grade quality will make biophysics for many hot targets possible. Bioinformatics play these days a huge role for a successful drug discovery program, and hopefully even faster in-depth interpretation of biophysics, biochemistry, and structural data will make our work easier. And of course, exploit screening synergies, like in focused DEL around identified hits or in silico guided merging of HTS and fragment hits. So there are different screening technology, all work great, but every screening technology has its advantage and disadvantage. It would be great to try to combine the advantage of each of the available screening technology in order to develop faster, very potent small molecules. I would like to thank you all. I enjoyed this webinar a lot. I hope you enjoyed it as well. And I'm looking forward now to the Q&A session and discuss with you biophysics more in detail.
Moran, Annika, Sun, extremely in-depth and wonderful presentation that I think will be of huge value to many of our folks in the audience. So we appreciate you sharing that with us. Thanks very much. To the audience members, if you have a question for our panelists, don't hesitate, send them on in. It's really simple. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit submit. We're going to try to get to as many of your questions as possible, so keep sending those questions in, and we're going to try to keep answering them. Bear with us for just a moment as we make the transition into the Q&A session. All right, everyone, thanks for joining us for the Q&A session. We hope you enjoyed the presentation, and we have a bunch of questions for our panelists, so let's get right to them. Moran, the first question is for you. One of our audience members would like to know, how comparable are KD measurements generated by SPR, MST, and ITC?
Yeah, that's a great question, and we really see that depends a lot on the system, on the target. We have systems where we have really great correlation between SPR, MST, and ITC, but there are sometimes cases that we see that in SPR we have measured higher affinity comparing to MST and ITC. This is mainly related to the fact that when we measure in free solution, we need to reach equilibrium, and for picomolar affinity that can take almost 24 hours, and therefore the affinity measured by fitting kinetic using SPR are significantly higher for very high affinities.
All right, thank you for that. Annika, we have a question for you. One of our audience members would like to know which technologies are the most suitable for fragment screening, and what are the throughput of MST and SPR?
Yeah, really good question. So there is not one technology that is most suitable for fragment screening. It might be either MST or SPR that they are using. So what we usually suggest is to do an assay setup with more than just one technology to see what works best in the assay setup, and then use this technology. In terms of throughput, and then of course you have the second or third technology also for the hit validation. In terms of throughput, so on average it's like 1,000 fragments per day. That's really an average value that might differ between an assay setup for MST or SPR, but that's the average we expect. Yeah.
All right, thank you, Annika. Ling Bing, we have a question for you. An audience member would like to know how good are biophysical data generated by MST and SPR? How do they correlate with classic cell-based degradation assays?
Thank you very much. That's a very good question. So for biofunctional molecule, actually we are have more interest on the binding affinity, which could be detected easily by the MST or SPR technology. And for the binding assay, we also have two different levels. One is the binary binding, the other is ternary binding. So for the data correlation, I do think the ternary complex formation assay is better associated with cell-based degradation assay data, especially for the alpha value I just mentioned, which is a good indication of cooperativity, and that's also a very good characteristics of a good degrader. Thank you.
All right, and Annika, we have a question for you. How long does fragment-based drug discovery take?
So we actually offer here 3 building blocks, and we strongly recommend to do an in-house protein production and protein QC, because we often experience that if you have a really high quality protein, then your FBDD is more successful. And this first protein production, protein QC building block takes approximately 6 to 8 weeks. The screening itself, which covers assay setup at the primary screening and hit validation with orthogonal validation and KD ranking, takes usually 3 to 5 weeks. In a subsequent step, there is also probably structure determination, structure-based drug design. This takes at least 2 to 6 weeks.
Okay, thank you for that. Sorry, Moran, our next question is for you. How do you typically decide which technology you prefer to use, MST, SPR, or DSF?
That, of course, depends, first of all, on the scope of the project. So if our partner is mainly interested in binding kinetics and not only binding affinity, of course, we will go with surface-based technology as SPR. And using MST, we measure binding affinity in free solution, having KDs as a readout. And there we have a lot of success with very different proteins. So, for example, for complex, we use in many cases microscale thermophoresis or TRIC. For antibodies, the first choice for us is always SPR. And DSF we typically use for protein to see how small molecular ligand affect protein thermal stability as a first try.
All right, thank you for that. Annika, back to you. How do you develop a fragment to lead molecule in the absence of structural information?
Yeah, so if there is no structure generation possible using X-ray crystallography or NMR, you can use an activity-led fragment to lead development. So to read out the activity of this distinct fragment on your protein molecule, and this will guide you through a more potent structure step by step.
All right. Moran, back to you. One of our audience members says, thank you for a great presentation, which I agree with. That was great. They say you presented multiple cases where the MST and SPR affinity values are tenfold different. Which value would you use? And are the values determined by MST or SPR?
Yeah. First of all, the reason why we need orthogonal validation, since measurement performed on surface can differ from measurement performed sometimes in solution. And we have discussed a bit, was actually the first question, that especially we observe very high affinity in SPR, and if you measure just a short incubation time in solution, you just get lower affinity. For me, I think that more important than absolute affinity is to compare what we see, for example, that if for a wild type we observe a certain KD in MST, and for a mutant we will see tenfold lower affinity. We will very likely going to see the same ranking in SPR, but the total affinity can be different. But we think it's important to have a scope and to be able to use orthogonal method in order to get the full picture. So if we see similar trends, even if the absolute affinity are different between different methods, as high TC, MST, and SPR, which based on very different technological backgrounds, then we can be convinced on our scientific conclusions.
All right, thank you for that. Lingbing, we have a question for you. An audience member would like to know which biophysical method is most suitable for the characterization of bifunctional small molecules.
Thank you. I think for the biophysics methods used to characterize the bifunctional small molecules, there are many ways, like the MST, the SPR, ITC, or even the mass spec-based technologies. So from the screening purpose, I think the MST and the SPR definitely stands out with a good affinity value as well as the throughput. Thank you.
All right, thank you for that. Moran, we have a question for you. Which labeling approach is most promising for challenging and buffer-sensitive drug targets in MST?
Yeah. So of course that there are different labeling strategy for MST. You can use covalent labeling approaches, labeling primary amine or cysteines, but for very different drug targets, we made a lot of very good success, and we are very successful using site-specific labeling. So I'd say the site-specific labeling is truly big advantage. It's very important to consider, but where is your fluorophore located on the protein? Since you do not want to have it too far away from the binding site, but not too close as well.
All right, that makes sense. Annika, we have a question for you. An audience member would like to know, are your DEL hits validated with the on-DNA compounds, or do you resynthesize the compounds off DNA?
Yes, that's a good question. So in this particular experiment, we have used off-DNA compounds that were resynthesized. In principle, it's also possible to use on-DNA compounds in MST and SPR. That's also possible. In this particular case, we have used off-DNA.
All right, and looks like we have one more question for you, Annika. Sean would like to know, what are the size limitations for RNA protein when it comes to SPR chips?
Yes, this then depends what to immobilize. So if the RNA is, for example, relatively small, one could immobilize the RNA easily via biotin and let the protein flow over it. It depends really on the ratio between RNA and protein and on the total size. Because, of course, sometimes we observe a spherical hindrance if the complexes are getting really large. But this is then really a decision project to project wise that we have to take.
Okay, and looks like we have one last question for you, Annika. Our instructor would like to know, is it possible to validate your DEL hits using the on-DNA compounds, and which methods would you choose?
Yes, we had a similar question already, and thanks for the question. So it's possible to use MST, and we could also use SPR for these on-DNA compounds, so both is possible.
All right, great. Next question for Moran. Moran, Vanessa would like to know, she says, first off, very interesting seminar. And for MST measurements for one target, like NLRP3, different MST are used for different molecules. Is it relevant to compare KD with different experimental MST parameters?
Yeah, so for MST, depends, as we know, on size, charge, and solvation entities. So depends where the molecule binds. It can cause different conformational change. So the MST response can give us some information. On the other hand, we need to say in many cases, different in MST response will lead to, or different in binding mode can lead to different MST response. But in order to understand exactly why, for example, a certain binding pocket gives us a certain response while binding to another pocket gives us a different response, it's very difficult since MST depends on different parameters. So it will be necessary to run other experiments and other orthogonal methods to understand and to find why a certain MST response was observed.
All right, thank you for that. And with that, it looks like we've come to our end of our webinar. So wonderful discussion, everyone. Thank you all very much for your time. I'd like to remind everyone that the webinar will be archived on the GEN website at genenginews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues who you think might be interested. we always recommend that. I'd like to thank Moran, Annika, and Lingbing for their amazing and in-depth presentation. And I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to Wuxi AppTec for sponsoring this webinar. Hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe, healthy, and get your vaccines, please.